Welcome to Chapter 12, Regulation of Gene Expression in Bacteria and Bacteriophage. The purpose of this chapter is to tell us stories about how genetic systems have been dissected. There are three main stories, and if you learn all three, you should be in a very good position to do well in this chapter. The principles within the first story, they help you with the second story. And once you learn those two stories, the final story really isn't that difficult, although it may seem to be from the outside. If your goal is to learn how things work at the molecular level involving genetics, then these stories are paramount for you to understand. Before we can begin with the first story, we need to put in place some housekeeping items. The next 15 or so slides provide the framework necessary upon which these stories are built. DNA by itself is pretty inert. It can't do much. That's the first thing to take away. The DNA does carry information for making RNA and then for making proteins, and it also carries information controlling access to itself, in addition to many other functions that are not important in this chapter. The most important thing for you to realize with slide number 1 is that there's always going to be some interaction between the basis of the DNA and the proteins that interact with it. All genes, no matter where they be, have to be activated at molecular switches known as promoters. These promoters bind proteins that activate genes, the most important being RNA polymerase, without which there would be no messenger RNA or any other type of RNA inside the cell. The principles which we're learning here in these few slides will also apply to the chapter on gene regulation in eukaryotes, as the interaction between proteins and DNA basically follows the same principles. Let's begin with a general observation that certain genes in bacteria are needed continuously during the life of that cell, and these are transcribed in a continuous fashion. Therefore, that type of transcription is called constitutive. That means never turned off, always transcribing the genes at the standard rates. Then we have those genes which are only turned on in a time of need. So those are regulated, and they're known as regulated transcription or facultative transcription. With respect to transcription, we're looking at two parameters: the initiation event, which transcribes the gene, and the quantity of transcription, i.e. the amount of transcript produced. Both of these parameters will have an effect on the amount of product produced. We may have an idea of what gene expression entails: initiation, elongation, termination of transcription. Then we have messenger RNA. Then we have the initiation, elongation, and termination of translation. In many cases, the protein product may be post-transcriptionally modified before it goes and does some kind of task for the cell. Any one of these steps could be regulated. But for most genes in bacteria, transcriptional regulation is the most common, whereby we're looking at the priming of the initiation site, the promoter, on the gene by RNA polymerase. Since RNA polymerase and the initiation factors are proteins, they must be able to determine which gene needs to be activated and when. There must be some type of interaction between the DNA binding proteins and the regulatory sequence of DNA. And that's where we start our story. Table 12.1 lays out the general groundwork for the first story and the second story, and tidbits of information thereafter. So we're going to take a linear dive through all these subsections of genetic control. We'll be examining inducible transcription, where the genes are turned on when needed. In contrast, we'll be looking at repressible transcription, where the genes are turned off when they're not needed. Thus, the default situation in the first example is the system is off, and in the second example, the system must be on. We also have some specialized cases of how to prevent a gene from continuing to transcribe should it be activated by another mechanism. we further look at how messenger RNA itself can be intercepted and destroyed before it's actually translated. And then we'll look at translation blockage, where the system intercepts at a later point the decision to make protein. When factors bind to DNA, i.e. when proteins bind to DNA and their binding reduces or eliminates transcription, we call that negative control. And these proteins that achieve this negative control have a label. They're called repressor proteins, and their job is to prevent transcription. On the contrary, other proteins that bind to DNA activate transcription, and those are called activator proteins. Let's examine some characteristics of repressor proteins. So repressors represent a very broad category of regulatory proteins. As we just mentioned, they have a negative control over transcription. These proteins bind to DNA, normally at places that have a similar label. These sites are called operators. They may be just upstream of the transcription start site. They may be at the transcription start site, or they may be somewhere downstream of the transcription start site. But the most common place you find them is around the promoter. What these factors do is basically bind to the DNA downstream of the RNA polymerase binding site. And they prevent the RNA polymerase from being able to move down the DNA into the coding section of the gene. In some cases, the operator and the promoter are one and the same. So the operator sits inside the promoter, and its binding of repressor prevents the RNA polymerase from binding. It's an exclusive condition, one or the other. You can't have both. The repressor proteins at the operator can themselves be regulated, controlled. So something allows them to find and bind to the operator, and something else may remove them from the operator and take them away so that transcription may initiate. Through evolutionary processes, these repressor proteins normally have two active sites. The first active site is a DNA binding domain, where the shape of the amino acids on the surface of the repressor protein allows it to bind to the operator and or other target regulatory regions of DNA. The other active site is known as the allosteric domain because it binds to another protein or a molecule that has an effect of causing a conformational change in the repressor such that the DNA binding domain becomes weakened or inactive, and that causes the repressor protein to disengage from its target regulatory DNA sequence. And that property of a protein is called allostery. The general principle of allostery allows proteins of different classes to behave in one of two ways. The first mode is, as we just discussed, the binding of something to the allosteric site causes the DNA binding domains to alter and the protein to release the operator or regulatory region. The molecule that bound to the allosteric site or the allosteric domain has a name. It's called the inducer because binding of the inducer causes the induction of the change in the protein. Conversely, you could have a different protein which binds something, and then that causes its DNA binding domain to become activated, so it can then go and bind to the DNA. So in that case, the molecule that bound to the allosteric site is called the co-repressor. Therefore, in the first example, the repressor has one of two choices. It can either bind to the DNA or bind to the inducer, but not to both. In the second example, the repressor has to bind to the co-repressor before it can bind to the DNA. Slide 12.1 illustrates this overarching principle. The association of a protein with DNA in both cases does not result in any transcription. Therefore, the presence of the protein factor, the repressor protein, has no transcription associated with it. In order to get transcription, you have to remove the repressor protein in both cases. In the top example, an inducer molecule actually lifts the repressor off the DNA, whereas in the second example, the dissociation of a co-repressor is necessary to remove the repressor protein from the DNA. Only then, in both cases, will messenger RNA or some type of RNA be produced from the transcription of these genes. Once you've managed to relate to the previous figure, then we can move on and talk about positive control. In positive control, transcription is accomplished by activator proteins, and they bind regulatory DNA sequences called activator binding sites. These proteins are normally designed to facilitate the binding of RNA polymerase and in initiating transcription. So they're helper proteins, not hinderer proteins. These ones hinder and these ones help. In order to recognize and bind to the activator binding sites, the activator proteins need to have a DNA binding domain. Once again, there's two modes of action for this positive control. In the first mode, the binding of DNA is initiated by the binding of something to the allosteric site on the protein. So the protein first binds its allosteric partner. That causes conformational change in the DNA binding domain and activates it this time. Therefore, it's able to bind to the DNA regulatory region. That binding activates transcription. With the alternative mode of action, the activator protein has its DNA binding domain actively blocked by some type of inhibitor that's binding to it. The removal of the inhibitor then allows the protein to bind to its DNA target and then activate transcription. Once again, a figure is useful. So in both cases, we do not have transcription yet because the protein is not bound to the DNA and recruiting RNA polymerase. So the absence of these activator proteins is why we call this positive control, because the presence of the protein is necessary to activate transcription. Whereas in the case of negative control, we had to remove the protein to allow transcription. And if you look carefully, you'll notice that the operator is sitting downstream of the promoter. So even if the RNA polymerase was to bind here, it could never get past this protein which is blocking its passage along the DNA. Whereas in this case, if you look carefully, the promoter is located at the point downstream of the activator binding site. So this actually sits upstream of the RNA polymerase, helps the RNA polymerase bind, and gives it a nice tug to get it moving down the DNA. In summary, in negative control, you have to remove the protein from the DNA. In positive control, the binding of the protein assists the RNA polymerase in transcription. When one examines the overall three-dimensional structure of proteins which interact with DNA, one finds a series of common motifs or patterns that occur. And these DNA binding proteins fall into three discrete buckets, those being Helix-Turn-Helix motifs, leucine zippers, and zinc fingers. At the individual level, how does one particular DNA binding domain determine uniqueness? And the answer is, it all depends on the contact made between the base pairs within the major grooves and the minor grooves of DNA and the amino acid motifs. So it comes down to the unique pattern and positioning of hydrogen, nitrogen, and oxygen atoms between the DNA and the regulatory proteins. The most common structure that's used is an alpha helix. An alpha helix is a right-handed spiral made by the protein that connects its atoms deep inside the DNA molecule. The manner in which these proteins have achieved these abilities to make contact with the DNA do vary amongst the three types. In some cases, we have a single polypeptide that folds to form two domains that bind to the target DNA sequence. In another type, the regulated protein consists of two or more proteins in a multimeric complex. In the case of the top one, we're looking at zinc fingers. In the case of the second example, we're looking at leucine zippers. If the two polypeptides are identical, we call that a homodimer. If they are different, made by different polypeptides, then that's called a heterodimer. With the case of bacterial regulatory sequences, most of them have something called an inverted repeat or a direct repeat along the DNA. Therefore, each polypeptide of the regulatory homodimer interacts with one repeat. The most common feature inside bacteria is the Helix-Turn-Helix protein motif, which consists of two alpha helices separated by a turn of amino acids. The two alpha helices interact with the inverted repeats, and the turn part of the polypeptide, the middle, just positions them in the correct orientation and spacing. Figure 12.3 shows a Helix-Turn-Helix protein making contact with its target DNA. And at the target DNA, we can see that the inverted repeats are key in allowing this protein to bind in either orientation. We are now ready to begin the first story. Some of you may be familiar with this, but are you familiar to the depth that's required in this chapter? So story A is of little red lactose operon. The lactose operon is a classic example used in nearly every genetics book, which illustrates beautifully the evolution of a system that we spoke about earlier in regard to something that's being inducible. So the operon is normally off and only turned on when lactose needs to be metabolized. It's an energy-saving protocol. This operon is under both negative and positive control with regard to proteins binding or being removed from its regulatory region. Whenever we find multiple genes under the control of a single switch, we call that system an operon. So in this case, we have 3 genes with one switch for transcription. Thus, we have 3 structural genes under the control of a single promoter. Evolution has compiled genes that perform a common function, that are needed in common amounts, to be switched on simultaneously or switched off simultaneously in order to regulate the process. Why have 3 different genes with 3 different switches when you can have one switch operating multiple genes? And that's what an operon is. The E. coli bacteria which reside in your gut have a preference for certain food types over others, mainly because it saves energy in utilizing some sugars than others. So glucose is the preferred source of carbohydrates for making ATP in E. coli. The reason being that glucose can be immediately sent down the glycolysis pathway, leading to the production of ATP. Alternatively, lactose is another sugar that can be used to drive the production of energy for bacteria. And the enzymes that allow lactose to be fed into the glycolysis system are made by the lac operon, and that is only turned on when necessary. So there's your inducible operon label. The decision to turn on the lac operon, or any operon that's inducible, depends on the presence of a trigger, and that trigger is called the inducer molecule or the inducer compound. And when it's available, the operon is turned on. When it's not available, the operon is turned off. Lactose is a more complicated molecule than is glucose. Indeed, lactose is a 2-ring sugar, consisting of one ring of glucose and a second ring of galactose, joined by a covalent glycosidic bond. Bacteria with a Lac plus phenotype can grow on media containing lactose as the only sugar because they're able to make the necessary enzymes for converting lactose to glucose. The first product of the Lac Operon is an enzyme called beta-galactosidase, and that breaks the linkage between glucose and galactose, freeing the glucose to enter glycolysis, and the galactose is converted by another enzyme into glucose, which also then follows into the glycolysis pathway. In order to allow the lactose to enter the cell, another enzyme called permease is also produced by the Lac Operon, and that then enhances the entry of lactose into the cell. During these maneuvers with lactose, a small tiny amount of lactose is converted by a side biochemical process to allolactose, and this is the actual inducer compound which will bind to another protein. Bacteria that are designated Lac- are unable to utilize lactose because of a deficiency in some of these pathways. This animation generated by the textbook company does a very good job of laying out the framework for the next number of slides that we have to go through. So what we're going to do, we're going to watch this first.
Regulation of gene expression. Prokaryotes. Organisms have evolved refined mechanisms to control the transcription of genes and optimize the use of available resources. This video presents one of the best understood examples of gene regulation in bacteria and demonstrates how the expression of genes involved in lactose metabolism and is modulated according to the metabolic needs of the organism. Negative and positive control. In response to environmental signals, regulatory proteins either promote or inhibit transcription. If a gene is under negative control, it is transcribed unless a repressor specifically blocks it. If a gene is under positive control, a regulatory protein is necessary to promote transcription. The Lac operon is negatively controlled. Its structural genes are expressed automatically unless inhibited by a repressor. Because the repressor is always produced, the operon is inducible. When lactose, the inducer, is present, it inactivates the repressor. The repressor is important for metabolic efficiency. It is better not to make enzymes for using lactose when lactose is absent. One of the Lac structural genes, Lac-Z, encodes the enzyme beta-galactosidase. This enzyme converts the disaccharide lactose into the monosaccharides galactose and glucose. The result of Lac structural gene expression is the cleavage of lactose into glucose and galactose. Galactose is later converted to glucose. What if a cell has ample lactose and glucose? It would not be efficient for the cell to be induced by lactose if glucose is already present. Another regulatory mechanism of Lac operon, called the catabolite activating protein, CAP, requires glucose to be absent for Lac operon expression. If glucose is present, the operon is not activated even when lactose is present. The operon cannot function well in the absence of bound CAP, and the presence of glucose prevents CAP binding. So, CAP exerts positive control over the Lac operon. Cis and trans acting sites. DNA sequences can affect other sequences either next to them, on the same chromosome, that is to say, at a cis acting site, or at relatively distant sites or sites on different chromosomes, that is to say, at a transacting site. If a DNA sequence has an effect over another sequence at a distance, a diffusible protein typically mediates the effect. If a sequence must be near another one to have an effect, it is said to be acting in cis. In the Lac operon, the operator and promoter are cis-acting because they function just upstream of the Lac structural genes. In contrast, the Lac-L gene can act in trans, producing a diffusible repressor that can travel to a sequence a great distance away. Lac operon. The Lac operon is a group of genes that ultimately produces the enzymes involved in lactose metabolism. The operon is made up of 5 parts. The 3 structural genes, Lac-Z, Lac-Y, and Lac-A, are transcribed into a polycistronic mRNA. RNA polymerase binds to the promoter, P, to initiate transcription, if initiation is not repressed. The repressor protein encoded by I binds to the operator site when lactose is not present, and prevents the initiation of transcription. Because transcription occurs unless it is shut off by the repressor, the Lac operon is said to be under negative control. Let us find out the function of each of the parts of the operon. Repressor gene. The repressor gene encodes the repressor protein. Unless lactose is present, this protein binds to the operator region of the Lac operon and prevents transcription of the structural genes. Promoter, operator. The promoter is the binding site for RNA polymerase and for the catabolite activator protein, CAP. The operator contains a binding site for the repressor protein. Beta-galactosidase gene. The Lac-Z gene encodes beta-galactosidase, an enzyme that converts the disaccharide lactose to the monosaccharides glucose and galactose. Permease gene. The Lac-Y gene codes for beta-galactosidase permease, which assists the transport of lactose into the bacterial cell. Transacetylase gene. The Lac-A gene codes for transacetylase, an enzyme whose role is still not completely clear. Lactose, the inducer. This diagram depicts the Lac operon, which contains the structural genes Z, Y, and A, as well as a promoter and operator. The Lac-L gene encodes a repressor protein, which contains binding sites for the operator and for a lactose molecule. The presence or absence of lactose determines whether the Lac repressor can bind to the operator site. When lactose is absent, the repressor remains active and can bind to the operator site. RNA polymerase can bind to the promoter region but cannot proceed past the blockade imposed by the repressor protein. No enzymes are produced in this situation. When lactose is present, enzymes are needed to metabolize the lactose. The repressor binds to lactose at the lactose binding site. When this occurs, the operator binding site is altered and the repressor is unable to bind to the operator site. RNA polymerase binds to the promoter and can proceed past the operator site. mRNA from the structural genes is produced and then translated into the enzymes involved in the metabolism of lactose. Mutations in the Lac operon. If a mutation in the Lac-L gene results in a repressor that cannot bind to the operator, the Lac-L negative cell cannot turn off the operon, and the mutation is called constitutive. When lactose is absent, a normal repressor would bind to the operator and block expression of the structural genes. However, the mutant repressor cannot bind to the operator, and the genes are expressed whether lactose is present or absent. In this example, the operator region is mutated. This mutation of the operator is called the Lac-OC mutant. The nucleotide sequence of the operator DNA is altered, changing the shape of its binding site. Because the shape is different, the repressor protein cannot bind to the site. Therefore, after RNA polymerase binds to the promoter, it can proceed past the operator to transcribe the structural genes. Catabolite repression. The Lac operon is under a form of control called catabolite repression. RNA polymerase does not bind efficiently to the promoter unless another protein, the catabolite activating protein, CAP, is present. CAP binds to the 5' end of the promoter at the CAP binding site. When CAP is bound, binding of RNA polymerase is enhanced. Thus, CAP exerts positive control by enabling efficient transcription when it is bound to the CAP binding site. Another molecule is involved in this process, cyclic adenosine monophosphate, or CAMP. For CAP to bind to the promoter, it must first bind to CAMP, and then the entire complex binds to the promoter. The presence of glucose determines whether CAMP levels are high or low. If glucose is present, the cell doesn't need to metabolize lactose for energy. The presence of glucose results in low levels of CAMP. Without CAMP, the transcription of the lac operon is inefficient. The lac operon is a gene cluster composed of promoter-operator region and 3 structural genes that function in lactose metabolism. These genes are coordinately regulated by a negative control system. In the absence of lactose, a repressor protein produced by the LAC-L gene binds to the operator region of the operon and represses transcription of the structural genes. If lactose is present, however, the repressor is prevented from binding to the operator and transcription proceeds. The importance of the repressor and operator in the regulation of the lac operon has been confirmed by experiments with mutants in which the operon is either constitutively active or inactive. The lac operon is also subject to positive control through the catabolite activating protein, CAP, which binds to a specific region of the promoter and then stimulates the binding of RNA polymerase. Polymerase binding is efficient only in the presence of a bound CAP at the CAP promoter site. CAP cannot bind RNA polymerase unless CAP is first bound to CAMP, however, and when glucose is in the cell, the level of CAMP is low. Thus, in the presence of glucose, the absence of CAMP greatly reduces transcription from the lac operon. This mechanism is known as catabolite repression and is energetically efficient because bacteria favor the use of glucose over lactose.
Boo! Wakey wakey, no falling asleep. I'm sure most of you failed to stay with that video, so what we have to do is make sure that you understand every component of that video plus a bit more. The first thing is to reiterate the biology. So if you are a bacteria in your own gut and you've just taken a meal of glucose, then that glucose will be used as the preferred source of energy. If you're taking a meal of just lactose, milk, and there's no glucose in your system, then the bacteria have no choice but to use the lactose. Interestingly, if the bacteria are given both sugars through a meal, they will prefer to use the glucose first, and then only when glucose runs dry will they switch over to lactose. Lastly, if neither sugar is available to the bacteria, they'll have to find an alternative mechanism of supplying them with energy, otherwise they'll die. Figure 12.4 shows you how lactose enters through the membrane of the bacteria, the E. coli. There's a protein called permease, and that protein is a channel for lactose. And once lactose has entered, it's a disaccharide, it's then attacked by beta-galactosidase. This enzyme splits the molecule at the galactoside linkage. The galactose is converted to more glucose, and both those molecules are then sent down the glycolysis pathway, as we noted earlier. The overall arrangement of this part of the bacterial chromosome consists of a left side and a right side. On the left side, we have the regulatory region. Attached to it, immediately on the right side, are the 3 structural genes. The regulatory region consists of 3 very important zones on the DNA. The first is the promoter that binds RNA polymerase. The second is the operator, called LAC-O, that binds the lactose repressor protein. And then on the far side, upstream, of the promoter is a site called the cap binding site. There's a small degree of overlap between these three DNA regions, but it's enough to allow transcription to proceed under certain circumstances. And that's what we need to learn. How do you control this very logical switch? In fact, it's more than a switch, it's a circuit. So the right-hand side of this region of DNA are these three structural genes, and they encode the enzymes beta-galactosidase, the enzyme permease, and the enzyme transacetylase. The LAC-Z gene produces the first enzyme, the LAC-Y gene produces the next enzyme, and the LAC-A gene produces the last enzyme. These three genes are encoded into a single messenger RNA, which is then later processed into three separate proteins. So whenever that happens, we use this genetic term. Poly means more than one, cistronic means genes, so there's more than one gene on the messenger RNA. Outside the LAC operon, therefore not part of the LAC operon, is another regulatory region, a gene with its own promoter. That gene is called the LAC-I gene, and what it produces is a protein. The protein is actually the LAC repressor, this very important decision-making component. This gene is always on. It's part of the housekeeping set, so it's constitutively expressed. The product of the LAC-I gene is actually 4 polypeptides. So it's a homotetramer. 4 polypeptides combining together to make two important zones on its surface. The first is the DNA binding domain, and that we mentioned earlier is able to bind in certain circumstances to the LAC-O region of the LAC operon. And then we have the allosteric domain that binds the inducer. And the inducer is a form of lactose that's produced through a biochemical process. Its real name is allolactose, but lactose and allolactose can be synonymously treated. A good representation of the actual arrangement of these components is indicated by figure 12.5. So in a general perspective, we can see the three genes, the three structural genes are here, with the Z gene followed by the Y gene and then the A gene. And just upstream of that, we have the regulatory region, to which we have an operator in yellow, the RNA polymerase binding site in orange, and the cap binding site in this lavender color. Then upstream of that, some distance, we have the LAC-I gene with its own promoter. If one is able to draw this freehand, that will be a very good asset to have. An annotated view of the regulatory region is depicted in panel B, along with the locations of various DNA sequences. So there's your -10 sequence, there's your -35 sequence, then upstream of that we have the cap binding site. Then we also have at the +1 site and up to +20 we have the LAC-O gene, which is the operator site. There's a short 5' untranslated region that contains the Shine-Delgado sequence for binding the ribosome, and then we have the first codon of the LAC-Z gene. Since the LAC operon is silent when there's no lactose in the cell, or when there is lactose but there's also glucose, glucose is given priority and the LAC operon is shut down. And we're going to see how that mechanism works at the protein, genetic, and biochemical levels right now. So when no beta-galactosidase is produced, there is no allolactose inside the cell. And the LAC repressor protein, remember it has 2 binding sites, it binds only to the LAC-O on the DNA, therefore blocking RNA polymerase from being able to get through that region of the regulatory zone. Once again, when something is blocking the DNA by binding to it, we call that a negative control system. The all-important protein is the Lac repressor. Here it is with two binding domains. On top we have the DNA binding domain, and on its sides we have the allolactose binding sites. In this pictorial, because lactose is absent, there is no allolactose, therefore there's no binding to the allolactose binding sites, and the protein is free to bind to the Lac-O region of the operator, thereby preventing any RNA polymerase that should bind to the promoter from proceeding to transcribe these genes. Only when lactose is present and glucose is not will the Lac-Operon be induced, woken up, and it'll start transcribing at a high rate. Because lactose is now present inside the cytosol, some of that will be converted to allolactose, and it's the allolactose that acts as the inducer. So it's going to bind to the repressor complex and pull that off the DNA binding domain, unblocking that segment of the gene. We have an illustration of that in figure 12.6b. The repressor binds the allolactose, and it no longer binds to the Lac-O site on the DNA. This gives an opportunity for RNA polymerase to bind to the promoter and then start making its way down across the three structural genes, producing their messenger RNA. We must stop you here in order to relay the different levels of transcription which are taking place. So imagine that transcription goes from 0% to 100%, and this is like a scale. And this analogy is to a dimmer switch, not an on-off switch. So the dimmer switch can be at 0% when there's no light, or it could be 100% where the lamp is illuminated at 100%. Or, depending on the mood, you can have the light down to any other value you like simply by sliding the dimmer switch to the appropriate location. This analogy works with the level of transcription. So when something is blocking the DNA, i.e. something is bound to the operator, the repressor protein for instance, then the level of transcription is down in the single digits, a few percent at most. When the repressor protein is no longer bound to the operator, as in this case, then the level of transcription is a bit higher, maybe 10 to 20%. That's it. So it's not going to be anywhere close to 100%. So this is called the basal transcription level. So only a small number of messenger RNA molecules per unit time are produced compared to when the genes are activated at a much higher rate. So basal transcription itself is not sufficient to generate enough messenger RNA to start metabolizing the available lactose. Then the question is, what causes this Operon to transcribe at very close to 100%? And the answer is, when glucose is present, another molecule called cyclic AMP or CAMP is low. So it's like a seesaw. Just imagine that. So it's a very complicated association between glucose and CAMP. But just remember a seesaw. When glucose levels are low, the levels of cyclic AMP inside the cell will be high. When glucose levels are high, the levels of cyclic AMP will be low. So if we're saying that in this cell, glucose levels are high, then cyclic AMP levels will be low, and cyclic AMP is a partner that the cap protein requires in order to bind to the cap site and assist in increasing the level of transcription up to about 100%. The positive control aspect is therefore initiated by the cap cyclic AMP complex. And the negative control is exerted by the repressor. When glucose is present inside the cytoplasm, the cap cyclic AMP complex does not form because there's not enough cyclic AMP to combine with cap. Therefore, there's no assistance provided to the RNA polymerase, and there's only basal levels of transcription. Another genetic term called catabolite repression is applied to this scenario. Thus, in example C, when we have lactose and glucose available, we're only going to get low levels of transcription of this operon, not enough to begin to break down the lactose into energy. So glucose is still the preferred source of energy. And you can see the situation. The repressor protein is floating around freely, bound to allolactose. It can't bind to the operator. The operator is not blocked. However, the level of binding of RNA polymerase to the promoter is very low. As we said before, about 7 to 10% of the activity of this transcription takes place in that scenario. Scientists have determined how the cap cyclic AMP complex interacts with the cap binding site and the maneuvers that it performs to encourage the binding of RNA polymerase at these very high levels. It does so by bending the DNA almost 90 degrees and therefore docking the RNA polymerase onto the Lac promoter at the perfect position and then giving it a push so that it can continue to transcribe the gene. Once one RNA polymerase has been pushed along, another one is recruited and another one, just like a train of RNA polymerases. Like most molecular systems, nothing is permanent. It's the example that we give you when the door is shut. It's mostly shut, but every now and again it flips open because of molecular energy and motion. And when a door is open, it's mostly open, but it flips shut every now and again as it's vibrating. The same thing happens in the case of the repressor and allolactose. They are bound to each other, but they're still vibrating with molecular motion. And every now and again, the allolactose falls off the repressor, and the repressor makes its way to the operator and blocks it temporarily, but not enough. The reverse is also true. When the repressor is bound to the operator, it's bound there not permanently, it can fall off because of molecular motion. And when it falls off, an RNA polymerase, which happens to be sitting behind it, can escape down and transcribe the genes at a level of 1 or 2%. We have learnt a lot about the way the Lac Operon functions by studying mutations of various aspects of its control regions and structural elements. Table 12.2 summarizes both the locations within the operon, the product or function, and importantly, it goes on to talk about some of the mutations that can exist for that region. So let's pick one. Let's go for LAC-A. So LAC-A is the gene, the last gene in the Lac operon. So it could be LAC-A is fine, or LAC-A is missing. So there's no product produced from the LAC-A gene if it's mutated itself in its sequence. Then for the LAC-O, we have a mutation called the LAC-O constitutive. That means the operator is unable to bind to the repressor because the sequence of bases at the operator has altered and is no longer compatible with the fingers of the repressor. We will mention all these mutations as we proceed into this section of the slides. One thing to understand before we begin any type of mutation analysis is this. E. coli is a haploid organism. It has one chromosome. But under certain conditions, we can introduce extra genes on a plasmid and pretend that it has a diploid constitution. So the plasmid carries a copy of the lac operon and its genes, so does the chromosome. So this is a situation akin to being diploid. But because only a tiny fraction of the chromosome has a corresponding second piece of DNA, we call those partial diploids. And they can be produced by conjugation between a plasmid called the F factor for lactose and a bacteria that is missing the plasmid and it only contains its own chromosome. Regardless, these mutations will allow us to experiment with different types of lac defects, and when these experiments were first performed by scientists, they found that the lac minus mutations fell into two complementation groups, and they now correspond to lac Z and lac Y. The partial diploid analysis uses this convention to identify the presence of the different genes and their mutation status. The important thing is that the F prime side talks about the genes on the plasmid. The slash divides the plasmid from the chromosome. So this side we have the condition of the lac operon on the chromosome, and on this side we have the DNA of the plasmid. The plus and minus symbols are standard genetic symbols. The plus means everything's normal, not a mutant, and the Y stands for that gene has a mutation within it that can affect its product. In this example, we see that the plasmid has a broken Y gene, whereas the chromosome has a broken Z gene. But overall, when you get expression of these two pieces of DNA, because everything else is perfect, including the promoters, the system will work in a normal way. So when lactose is present and glucose is absent, the production of these genes will take place, and the Z gene from here will produce a product, and the Y gene from here will produce a product. Therefore, complementation will take place, and we'll get a full set of genes for breaking down and utilizing lactose. One needs to understand the syntax being used before proceeding to subsequent slides. It makes no sense if you don't understand the system. So whenever you see something like this, we're just looking at the bacterial chromosome. There is no second piece of DNA inside that cell. We can see here everything has a plus sign. So for row 1, everything is normal, and we call that the wild type lac operon. So what happens? What do these symbols mean? They just tell you what happens in those biochemical scenarios. So when lactose is present, then we will get the beta-galactosidase produced. When there's no lactose, then we won't get beta-galactosidase. Likewise, this section here refers to permease. When lactose is present, permease will be produced from the Y gene. When there's no lactose, then there'll be no activation of the operon, and therefore there will be no permease inside the cell. The last example, number 4, now brings back the plasmid. So we have the DNA from the bacteria, and then we have the DNA from a plasmid. And the two will complement each other if they can, and that's indicated there. Certain mutations that we'll look at in a few moments will permit the lac operon to remain permanently on. It can't be shut down. So that is now non-responsive to the presence of lactose or not. And those are called constitutive mutants because the DNA behaves in this constitutive fashion. Other mutations will affect the switches that determine whether the circuit should be on and off, and they can make the system unresponsive to the presence of lactose, and therefore they mimic the lac minus phenotype. Incidentally, genetic mapping techniques of constitutive mutants were instrumental in localizing the lac O and the lac I regions to these mutants. The two sites that Jacob and Monod discovered through their experimental analysis happened to be the Lac-I gene itself, the one that produces the repressor, and the target of that gene, one of the targets, the Lac-O, the operator. One mutation affects the production from the Lac-I gene of a protein, the repressor. Now, once the repressor is made by the ribosomes, it's free to wander around the cytosol of the cell, binding to any operator that it comes across. So in that case, we say that the repressor is transacting, whereas the DNA elements are tied to the structural genes physically. So if one operator is present on the chromosome, it's only tied to the structural genes next to it. It can't wander and bind to the plasmid structural genes. And on the plasmid, the operator there can only influence its genes and not the genes on the chromosome. So in that regard, we call those elements cis-acting. They can only affect the same chromosome on which they reside. So this is a general rule of genetics, that elements or regions of DNA are cis-acting, whereas proteins or factors are transacting because they can move in various directions. Mutations in the sequence of the operator lead to, in some cases, the repressor not being able to recognize that region any longer and thus unable to bind. Therefore, repressor can never bind to that particular operator. So those operators are called OC for operator constitutive. And these mutants are useful in then analyzing the behavior of the repressor. Without a protein blocking the promoter and the RNA polymerase, these genes can be transcribed either at low level or high level, depending on positive control that we spoke about earlier. But what cannot happen is that the expression cannot be repressed down to the 1 or 2% that we spoke about earlier. By making partial hybrids using different types of chromosome and plasmid combinations, the scientists were able to show that if you had a piece of DNA in which the normal Z gene was tied physically on the same piece of DNA to the OC mutation, and at the same time you had in the same cell the wild type operator tied to the LEC-Z mutant gene, this led to this piece of DNA being unable to be blocked. So the cell would express Z gene regardless of the second chromosome. And we know the reason why. And when they produced an alternative arrangement where the OC mutation was attached to the Z minus gene and the normal operator was adjacent to the Z plus gene, they found that expression of these genes was under normal control because the repressor could still bind to the operator here and control the activity of this particular gene. So that led to a conclusion that the operator sequence on the DNA is cis-dominant and that it only influences the genes downstream of its physical presence. In this example, there's a normal wild-type operator, and here we have an operator that carries a mutation in its sequence, and that can no longer bind the repressor, regardless of which state the repressor is in. See if you're able to now decipher the genotype and relate it to the phenotype, and then the types of mutations that can exist. Let's turn our attention to the Lac-I gene. If the Lac-I gene on a piece of chromosome in a haploid cell is mutant, it cannot produce any repressor. And if it doesn't produce any repressor, then nothing can bind to the operator, and therefore the structural genes will be expressed at whatever level. In a partial diploid, if you have the Lac-I gene on one chromosome, and that carries the Lec-Z and Lec-Z plus in this configuration with the Z minus and the Y plus, And on the other chromosome, you have the mutated Lac-I gene, and that one carries the converse relationship between Z and Y. What would be the result? Well, you will get normal expression of both genes. Why? Because even though this Lac-I gene is mutant, this one is normal, and this one will produce more than enough normal repressor, not just one unit, but tens of units, hundreds of units, and they will diffuse and bind to the same chromosome or the opposite chromosome and start encoding the Z plus gene from the first chromosome and the Y plus gene from the other chromosome. Z plus and Y plus. So this is an indication that the regulatory protein, the repressor, is transacting, that it can wander and bind to any operator that's available and compatible. So there's a diffusing and interacting component to these proteins. In this example, the Lac-I gene is once again mutant, but instead of not producing any protein, a null mutation, it produces a protein which has a defective DNA binding domain. Therefore, it's unable to bind to the Lac operator, but it can still bind to the allolactose. Since the Lac-I gene has a protein with two active sites, there can be instances where a mutation in a different part of the Lac-I gene will not destroy the DNA binding domain, but it will interfere with the allolactose binding site, the allosteric site. In that case, we see something else. We call that component, the protein, a super repressor, because it cannot be pulled off the normal operator under any circumstances. There's no way that allolactose can bind and pull that repressor off the DNA. So in that case, the term that we use for the gene mutation is IS and not I minus. And the IS stands for super repressor. So there's three types of mutation for the I gene. The wild type is I plus. The I minus simply means it can't bind to the operator. And then the IS means that it can't be pulled off by allolactose. The mutation in the Lac-I gene is indicated to involve the allolactose binding pockets. Therefore, allolactose could never bind, and this pathway is not available to this protein. The only thing it can do is it can bind to a normal Lac operator if it can find one. And once it finds it, it cannot be pulled off in the presence of allolactose. The result is that that particular segment of DNA will be permanently shut down, apart from the leaky example that we gave earlier, 1 or 2%. Mutations can happen to any piece of DNA, and the promoters are no exception. So mutations in the promoter come in two flavors. You can be normal, the lactose promoter could be normal, or it can have mutations which reduce or eliminate the binding of RNA polymerase, therefore affecting the transcription of the Lac-Z and Lac-Y genes in this example. But these mutations, they operate in the same way as previously. Those mutations are going to be cis. Table 12.5 brings back the different combination of metabolites that are present in different scenarios. So we should have all four permutations of the glucose and lactose, and then we can associate those with other elements that are present inside a cell and then explain fully what's going on in terms of regulation. This slide corresponds to one of these four conditions, and in this case glucose is absent and lactose is present. So in that scenario we can look it up. Glucose is absent, lactose is present. Therefore, when glucose levels are low, cyclic AMP levels are high. And that cyclic AMP will then bind to the cap protein and go and assist in attaching the RNA polymerase to the promoter. At the same time, the allolactose is also present inside the cell, and that will bind to the repressor and pull that off the DNA. So the combined effect is that you have positive impact on the rate of transcription here, and then you have a positive effect on the rate of transcription here. Therefore, you have high levels of transcription. Molecular analysis and breakdown of the chromosomes around and within the lac operon is now beginning to show its secrets. The operator is in fact overlapping with the promoter to some degree, the operator region. And the three distinct segments of the operator DNA sequence have been identified: O1, O2, and O3. The association of the lactose repressor protein with the operator DNA has now been deduced. So this side of the protein is where the allolactose binds. So this is the allosteric domains on the 4 polypeptides. And then we have the DNA binding domains here, and they fit the DNA at each one of those 3 O sites, O1, 2, and 3. In fact, the only way that the 3 operator sites are able to interact with the repressor is if the DNA forms a loop. Any mutations that prevent the formation of the DNA loop and the binding between the repressor protein and the DNA will result in a mismatch between the two. So in OC mutations, operator constitutive, the mutations are here, and you can see they will prevent this branch of the heterotetramer from binding. If you wish, there's a video that accompanies this textbook chapter that you should watch. Thank you.